Welcome back to Evolving, the podcast designed to help you strive, thrive, and optimize. I'm your host, Nita Jane, and today I'm joined by Richard Sprague, a software engineer and quantified self enthusiast. Richard has served as a high level executive at both Microsoft and Apple and worked for numerous startups. He co founded Ensombio, a personal health tracking app, as well as AirDoc, the largest AI based tracking company in China. He holds a degree in computational linguistics from Stanford, as well as an MBA from Wharton. Richard, thanks so much for joining us today.
It's an honor. I'm glad to be here. It's good to talk to
Likewise, I just wanted to ask you, how does your experience as a software engineer inform your approach to personal science and the quantified self?
you. Well, that's a good question. And as you know, there are a lot of other, it's an occupational hazard of being a tech bro, is you tend to think of the world in terms of like these computer algorithms. And part of the way that I got started was I was very early. I guess I was early enough in the personal computer revolution that that I saw like the potential. I started using my first personal computer back when Apple had was just becoming something that people knew about, and IBM PC and those kinds of things. And but I was a little bit too young to be the next Bill Gates or somebody like that. But I really appreciated the idea that anybody could get involved with computers. It was in those days like you could just get started by being just a normal person. If you read the manual and you work at it, you could do pretty impressive things. And a lot of software companies were started by people with no particular degrees or no particular relevant degrees, because a lot of this stuff was being invented as we go. But the software engineering part of it for me was relevant because it's pretty easy when you think about computers and you understand how how the hardware and the software unite, to start thinking of your body as being a type of hardware. And then you ask, like, what is the software? And then you ask, well, how can you manipulate the software? Because software is the kind of thing that you can change as much as you want and however you want. And I was just always intrigued by that. And coming to the next level, like by the time that the Human Genome Project started, I was really intrigued by the idea that, well, maybe we are just pieces of hardware and we can just start tracking our genes, like our blood results, like a whole bunch of things, just the same way that we would track, say, parts of a computer system. So it's kind of natural, at least it was to me, to move from thinking about like computers as hardware versus software and thinking about humans the same way.
Gotcha. What do you think about the complexity of human biochemistry and the microbiome compared to what you experience with computers? And like,
So that's a good observation. So I guess I feel like that general idea that there's some kind of analog between computer hardware and software and human hardware and software. It turns out to be a little bit misguided, because obviously the human hardware is way, 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 way more complex. And there are multiple levels too that aren't obvious to somebody who's just a software engineer, because it's not just about calculating every atom or every chemical that goes in and out of your body. We're part of a bigger subsystem of things that include things like your environment, your culture, your upbringing, a whole bunch of other things that go into it that it's not that easy to model.
absolutely. I've often heard the analogy used that our genetic code is sort of akin to a computer's hardware, and then epigenetics is sort of the software that runs on top. But even that's rather simplistic when you consider how expansive epigenetics really is, and just how complex metabolic mapping is, and how everything kind of interacts with one another as part of the exposome. It gets very complicated
Yep.
very quickly.
Yeah, that's right. And then part of the thing that intrigued me about the microbiome when I first heard about this, that there's another dimension there of, that most of the genes that are in and on you aren't even your human genes. So, it's a level beyond the epigenetics, because there's a whole bunch of other organisms that affect everything that you do. And we're just beginning to understand kind of what that whole system is about. But the one thing we know for sure is that it's extremely complicated. It's not just a program that an algorithm that you have to debug. It's way more complicated than
Definitely.
that.
Definitely. And, a lot of the time we hear that 10 to 1 ratio given of like microbial cells to human cells. It's interesting because when they recalculated this, they found a number that was closer to 2 to 1. But if you're looking at
Hmm.
just DNA bearing cells, if you kind of ignore the component of red blood cells because they obviously don't carry DNA, then you again get that 10 to 1 ratio of microbial cells to human cells if you're just looking at things that are carrying genetic information, which arguably could be more important just because those are kind of being transcribed and then translated into making protein.
Yeah, I'm not sure that's a super interesting observation that like just comparing cells to cells, because if you really care about genes and gene expression, then like how many cells it is doesn't matter much. And then even beyond that, every time you're constantly losing microbes, so it's not even clear to that it means anything to say that you've got 10 to 1 or 2 to 1 or whatever. It just depends on the time of the day. So like every time that you have a bowel movement, you're releasing a ton of microbes. But I think the more important thing is just the observation that uh, whatever the number is, it's way more than what we have as human DNA. And we have no idea what all those things are doing.
Very true. Just going back to your point about the diurnal variation, the fact that the microbiome does undergo shifts even within a 24-hour period, what does this mean for microbiome testing? Does this mean that we shouldn't be giving any too much credence to any one particular time point? And like, is it better to perform longitudinal sampling if we want to get a good snapshot of what's in our gut?
Yep. Yep, that's one of my big conclusions about things is that when somebody tells me that they did a microbiome test and they did just one, I always look at that with a big, degree of skepticism. I think you really need to do at least two, because what you'll find out is there's a lot of variance even between two of them. And then, like you say about longitudinally, you really need to look at the microbiome over time. And unfortunately, like with the gut microbiome, uh, you can only sample it, at the most a few times a day, but usually for most people it's once a day or every couple of days. It's much harder to sample, um, and you really have to look at it as just snapshots. The other thing about the gut microbiome is that you're only seeing like one part of the human microbiome. It's just the part that it's an important part, and it's the part that happens to be in the the large intestine. But there's a whole bunch of other things that are going on too that that are harder to access and yet might be just as or even more important. So it's just a big, complicated mess.
It's true. Yeah, we do use the colonic microbiome as a proxy, but seeing as though everything is kind of adjusted to that proxy, I guess it still does have some relevance, some meaning that we can derive from the results. I
Yep.
think one good thing to mention here is that when people receive microbiome testing results, that data is usually scaled to 100%. So then, if you are having shifts in bacteria, like you have 5% lactose ratio one day, 10% the next day, that doesn't mean that you necessarily gained 5% because this is, of course,
That's right.
scaled as percentage of abundance. What are some
Yep.
other pitfalls when it comes to interpreting microbiome results that a layperson might want to be aware of?
Yeah. So that's the sum to one problem, or it's called the compositional problem, and that's true actually of a lot of biohacking tests. Anytime that you've got something where you're not able to sample the entire environment, you have to take a subset of it. You're depending on the idea that that's first of all evenly distributed and that you're getting a representative sample, and that's not necessarily true. And because it's not necessarily true, then when you get a report from a microbiome company, it shows on a scale like you say, it's like they make it as a percentage, but you don't know, maybe they're grabbing this percentage here and on the first test and this percentage here on the second test. It's just really hard to be able to tell. But the other thing that say a layperson doesn't necessarily appreciate, because most microbiome testing doesn't give you this result, is that a lot depends also on how they sequence the microbes. And there's this concept of the number of reads that they take. And that can vary a lot from sample to sample. And each bioinformatics pipeline, used by different companies or different labs, have different standards for what they'll tolerate for things like error rates, or like how many times they're going to sequence it before they feel like they're pretty confident that they identified a particular microbe or whatnot. And so that read count, which usually is not reported, that can vary by 10x between samples. And depending on what it is, you could seriously misread whatever kind of results you have, because you might just be dealing with a sample that they're just not at all that comparable in terms of read counts.
Very
Um,
true.
but
I
there
work
are lots of examples like that.
True. I actually worked at a microbiome startup for a few months, and we actually sequenced gut and vaginal samples. And the gut samples, as you might imagine, had a much higher sequencing depth because there's a much greater diversity of bacteria there.
Yeah,
And the vaginal microbiome doesn't need that same degree of resolution because of its unique properties and composition. But then we also noticed that, like, depending on which laboratory we were contracting with, we would get different results when it came to the gut information, the raw data that we were observing. And a lot of the time, just the DNA extraction step itself would make a big difference in what's available
yeah,
to amplify. So, for example, some laboratories, they use something like bead beating, or they might use some sort of lysing agent, and that'll make a big difference in the bacteria that you can then later amplify later down the pipeline. So all these different steps, there's bias being introduced, and I think that's one challenge in being able to compare across studies, because everyone has their own methodology, everyone kind of has their own way of sequencing. So how do we actually make sense of like what's happening across studies when there is so much heterogeneity, so much
yeah,
variation?
that's
And
true. And it's yeah, no, that's a really good point. And even something like, so like the American Gut Project, which is arguably the most science-based and maybe the most lauded microbiome set of microbiome studies that's out there, or at least has been in the past. Um, their samples are shipped in the air. In other words, they don't even use a buffer or anything like that. So they collect the sample and they just send it as it is in the mail. And for how long it's been in the mail, who knows? And they recognized early on that that was a problem, and there was a difference between when they started the project and maybe buffers and whatnot were not well understood, or people hadn't standardized on a lot of these things, but they didn't want to change their protocol, because then it would mean that it was harder to compare early samples with later samples. And a lot of people don't recognize this, but one of the things that they did was, or that they do with their data, is they actually run it through a filtering algorithm, because they know that some microbes grow faster than others when they're just out of the body. And so they filter that out and they do some bioinformatics corrections on it. And they've got all kinds of evidence and studies that show that it's kind of okay, and it's certainly okay for the kind of conclusions that they feel like that they were able to justifiably reach. But when you realize that this is true, even for a marquee project like American Gut, who knows what kind of assumptions they're making, but behind all these other studies that you see. And you see studies all the time that say that find a link between this or that microbe and such, such and such like pathology. And it's just really hard to tell how much you can compare these things.
very true. Yeah, just going back to your point about buffering agents, I had once read in a systemic review that the choice of cryopreservative seems to introduce
Hmm.
the greatest bias when it comes to a sequencing protocol. And I guess, that really differs across the board when it comes to different projects and the way different commercial organizations and the way different labs in academia do their sequencing. So there's quite a bit.
Yeah, yeah, that's right. And now it's not hopeless. So so many reasons why I still think it's really interesting to, and I think everybody should get their microbiome sampled. But one thing that tells you is that if you're going to try to compare samples or compare like conclusions, you really kind of want to make sure it's the same lab each time.
Yeah.
And you want to make sure that that lab is using a consistent protocol, because comparing protocols from different labs it's just really hard to make those assumptions. But one other example of something that people don't necessarily think about, but I like, that I think is hugely important. Um, it's well known that Akkermansia is a microbe that plays a huge role in the butyrate production in the gut, and a bunch of other, like, really important factors in human health. Akkermansia is interesting because it's only found in humans, and there's only one strain apparently that matters. But I run into people all the time who are like, they do one test from some fly-by-night microbiome company, and it says they're low on Akkermansia, and they're like, oh my God, what can I do? What can I do? And now there's even a like after much effort, somebody managed to make a probiotic that apparently can restore Akkermansia.
Yeah,
Um,
pendulum.
I've not used it. I don't know how it works, but yeah, and some people say it's good. I don't know. I don't know anything about it, but I do know that from my own testing, using the same exact lab, when I take the sample and we call it an evacuation, and I sample it from a different part of the evacuation, I get different levels of Akkermansia. And so my advice to people who are low in Akkermansia is the next time you test, try to test from the outside instead of the inside, because the outside is where when it's going through the colon. That's where the Akkermansia live. They live in the mucin layer of the large intestine. So if you can sample it from the outside, you'll end up with much higher levels of Akkermansia than if you sample in the middle. Of course, that's ridiculous to have to say that, because what that tells you is that you can't trust any of your results if you're not, being consistent the way you're sampling. And I think that's an important point that a lot of people make. So online, I see a lot of people who are like all excited because they followed such and such advice, and now their Akkermansia went up, or they followed such and such advice. Somebody else followed the same advice and they couldn't get their Akkermansia to budge. And I'm like, well, there could be another reason here why it's not showing up on the test.
That's true. Yeah, because stool itself is not homogeneous, and Akkermansia muciniphila, like not only does it kind of reside in that outer mucin layer, but it's also really, really responsive to fasting times. So I guess also depending on maybe when you sampled, you could see some variation for
That's
that
right.
reason.
So there's just a lot of things you have to find seem to correlate with problems. And there are other studies that show that too high of levels of Akkermansia seem to correlate, with problems. So like we're not sure exactly what the optimal level is, but there probably is a level that's more optimum than others. But again, it's just like super complicated.
Yeah, it's interesting with akkermansia because it obviously does exert some beneficial metabolic effects in terms of being like inversely correlated with obesity and metabolic syndrome, things like that. But at the same time, a lot of the time, there is this argument that if you're seeing high levels, it's not the akkermansia that's causative. It's just the fact that there's maybe mucus overproduction, and that's why you're seeing this bloom of akkermansia. So it's also kind of
Yeah, yeah,
difficult to piece apart the cause and the effect sometimes.
yeah. That's right. Yeah, that's right. Yeah, I think you have to look at it as just one piece in the puzzle, and
Definitely.
look at like there are lots of different other aspects of health that you should be looking at, and you have to just look at this as one of them. Um, Another thing that that's important to me, we call it personal science, but a lot of this,
Yes.
one of my learnings over the years has been the idea that um, just because it says in some study someplace that such and such is true, it may very well be true that of the people in that study, most of them, turned out a certain way, and so that may very well be true. But if you were one of the outliers in the study, that means that study doesn't even apply to you. And in every study, in every clinical trial, there's always outliers. And most of the time, a clinical trial reports the median value, or it reports some kind of thing that is considered representative. But if you look more closely at the data, there's almost always plenty of examples of people for whom the conclusion doesn't apply, or maybe even the opposite. And if you're one of those outliers, again, it just doesn't, then that study doesn't apply to you. So to me, the core insight that I guess I've learned over the years is you really got to think about this as a personal science project where you can look at studies, you can look at data that is collected from other people as guidance. But at the end of the day, you're going to have to think about it in the context of your own life and what you found to be true for yourself, or just from your own observations about things. And you know, that's that's a tough thing to really understand and appreciate. But unfortunately, that's just the truth of dealing with something as complex as human biology.
Very true. You compiled a lot of your personal findings into a document called The Personal Science Guide to the Microbiome. And I
Hmm.
guess, kind of
Hmm.
going back to your point about how a lot of people might be outliers, could you talk to us a little bit about the importance of N of one studies and this self experimentation for kind of figuring out what works for your individual biology and kind of how to kind of how to apply your findings to improve whatever it is that you're trying to optimize.
Yeah. Let's see here. Maybe I can show you. Maybe I can show you a couple of charts of that work on this. If we try that, let me see if I
Absolutely.
can share this. Because I've got a couple of things that that might be interesting to people. You mentioned the personal science guide to the microbiome, and that's kind of where I try to keep all of my latest, like understandings about things. I try to keep it up to date, and I'm actually working on a much better revision of it as I go through time. But there's a lot of charts and data in there that are helpful, including the ones that I'll show you right now. But let me see if I can share. Let's see. I click this thing here, and I'll put this one. So here, let me just show you a couple of quick things here that I think are interesting. Um, I think you can see this, right? Let me see if I can
Yes.
I turn that into a full screen thing? So kind of here was one of my learnings about kombucha, which everyone says is good for your microbiome. So I decided to try it, and I did again longitudinally. So I sampled every day, and in fact, for a number of years, I sampled my gut microbiome just about every day. My oral, no nasal, and skin microbiomes also like very regularly. And so I have a ton of data which I've been analyzing, and it's all written up in that ebook. Um, but one of the things that I found when I was looking at kombucha, for example, in this case, I tried to drink a ton of kombucha for a week and see what changed. And these blue dots here indicate days when I drank 3 bottles a day of this popular brand of kombucha. And as you can see, like I don't know if you can tell anything about my gut diversity here. You're a better pattern recognizer than I am. I didn't really notice much of a difference. Um, you could kind of, if you really wanted to see some kind of pattern, I'm sure you could come up with some kind of pattern. But I think that my conclusion was that it just didn't do a whole lot. I did find out that when the makers of this kombucha add a particular form of it's a bacillus microbe, and I did find out that sure enough, I was able to see some of that in the gut microbiome. So so there was some indication that this microbe had made it through my system. But again, did it do any good? I don't know. Um, and my guess is probably it didn't do any good. So I probably didn't. Probably didn't gain much. Similarly, I did the same thing with probiotics, and people are always like really excited about probiotics because it's a big, tens of billions of dollar a year industry. And if you read the labels on these supplements, they'll give all kinds of reasons why this is the best thing ever. And many of them are also able to point to studies of various quality that show that it does this or that. When I found out just from doing my own testing, and this is like kind of a really good chart of what I discovered. The red line here is a period, I think it was 10 days, where I took a pretty powerful probiotic that was given to me by the manufacturer. And I tried to change my bifidobacterium levels. And as you can see here, I don't know, like, did it really change? I don't know. I mean, again, if you really squint a lot, you might be able to say, oh, yeah, well, there you go. You were kind of low and then it went back up again. But I don't think so. I don't think that's any proof. Now, what's interesting about this chart is there's a period here where it's kind of off the charts high. And, that's what you care more about is like, why is it so much higher over here? And it turns out that this is during a period where I was eating a different kind of diet. And so one of the things that I learned as a result of this is probably diet affects the levels of bifidobacterium more than anything else. Um, so that's an example of the kind of thing that you can sort of like. I'll show one more example, and we can talk a lot more if you want to. But I also tried kefir, and in this case, I did find pretty obvious evidence that it works. Um, the blue dots are days when I drank kefir, and the rest of the time I did not. Now, I was making my own, and that's an important side note because I'm not sure that the kefir that you buy at a grocery store is necessarily going to have the same results. I've talked to people who claim various different kinds of results after drinking store-bought kefir, but it's my belief that the microbiome inside you relates somehow to the microbiome that's in the environment. So there's air, there's microbes that are in the air, there's microbes that are in the food that you eat, and there's kind of a buffer going on where your body is either in tune or not in tune with your environmental microbes. And one of the things that kefir does, if it's made in your home, and it's very easy to do, is that it kind of, you can think of it as almost like it's absorbing the local microbes and digesting it in a way that's more palatable to you. And so I found that it definitely leaves a mark. And I did a number of experiments like this that just sort of that I think can show uh, yes or no for me whether these things had much of an effect. But again, it might be the case that for you the results would be different. It's just kind of hard to tell. That was kind of my conclusion. But these are a couple of things that, at least in my case, I think I'm pretty clear that pretty clear evidence of what works and what doesn't work for me.
That's brilliant. Thank you for sharing that personal data with us. Just kind of going back to your point about the bifidobacterium, it's interesting that there are some hunter-gatherer groups in the world, like the Hadza, for example, that don't seem to possess any of this genus. Do you think there might be a bit of a problem in saying that bifidobacterium is a probiotic group and that maybe there are certain populations that don't require this for wellness? Is it possible that we're using these categories a little too broadly, or that maybe it's relative what is a probiotic in one population or another?
Yeah, that's a good point. I think that's right. I think people get a little carried away for thinking that certain types of microbes are quote unquote good and some are quote unquote bad. Um, it's hard to say. I don't want to be too pessimistic here and say that because there's this counterexample or that that it doesn't matter. Um, there's a lot of study that's been done on bifidobacteria, especially in Westerners or people on a Western diet, that seems to show that it's important. And so I wouldn't necessarily want to throw that body of evidence out just because some Hadza results show up differently. Um, but yeah, it's just really complicated. When I worked at Ubiome, we had collected a lot of data from a lot of people. And some of this was never published, but one of the things that I was personally really interested in was bifidobacterium levels. And unfortunately, the company went out of business, and we weren't able to publish some of these results. But one of the things that was pretty striking to me, it's one of the discoveries that we made, is that we were able to look at people, especially women, by age, and show that there's a major difference in bifidobacteria levels between women under, say, around age 30 and those over age 30. And now, is that causal, or is that an effect? I don't know. But like, it's those bigger picture ideas that I think are things that are kind of useful to look at. And it was pretty clear that the women who were under 30 were healthier. And was that because of the bifidobacterium levels, or was it was the health causing it? I mean, it's like hard to tell which direction the causality goes. Uh, and then it's also not clear that if you just magically gave a whole bunch of bifidobacteria to the women who are over 30, would that matter? But it does tell you, if nothing else, I think it hints that if you just go based on an overall study that shows that it's good or bad, that's hard to interpret if you don't know other ways that that study was broken down by, like in this case, age, sex, or even, we didn't have the kind of resolution where we could break it down by, by the part of the country or diet type or, lots of other things. So, who knows what else is going on. But still, like in the aggregate, it was interesting that there was this overall general trend, and that might relate to something that produces some actionable consequences for people.
Definitely. I do think it's so important to remember that, the environment in which we find ourselves very much informs what will or won't be
Hmm.
beneficial. And as you're saying, across Western populations, bifidobacterium are considered really quite beneficial. And I think even when it comes to IBS remission, if you're trying to do FMT to treat that, a donor enriched in bifidobacteria usually leads to a higher rate of remission versus having a donor that doesn't have
Hmm.
that much bifidobacteria to transfer.
Hmm.
Just talking about the environment once more, you did notice that you picked up a strain of seaweed polysaccharide digesting bacteria that's usually just found in people of Japanese ancestry. Talk to us a little bit about that, and like, what do you think that means for the malleability of the microbiome, and how much of it we can actually have the power to change potentially?
Yeah, in fact, that was that was one of the intriguing results that got me interested in the microbiome in the first place. So, I was an early user of 23andMe, and I've always been interested in genomics, partly because of that, that human hardware versus software kind of thing that intrigued me. And one of the disappointing things about, the genome, the human genome, is that you can't really change it. So like you're kind of stuck with whatever you're stuck with. Now there's epigenetics. There are other ways of maybe modifying or like thinking that your genome isn't as fixed as it might appear. But the microbiome was the enlightening part, the observation to me that you can make major changes to it. And the thing that really got me interested was this study that was done at I think it was the University of Washington, St. Louis, Washington University in St. Louis, where they looked at a representative sample of people in the United States versus in Japan, and they found out that there's this one particular microbe that is that they found in Japanese people, but not in North Americans. And after I looked at my initial, the first couple of times that I did my own microbiome test, I found that I have this microbe, which was kind of cool. And because I'm North American, you'd think that I wouldn't have this, but it turns out that in my younger days, I lived for about 10 years in Japan. So I was exposed to the Japanese environment, and I spent a lot of time eating, obviously, Japanese food and whatnot. And so it appears that I picked up this microbe somehow. So I'm an exception to that rule that says that North Americans, don't have this. And it's possible, and I think that's what kind of set me along the way of thinking about how important the microbiome and your diet and environment might be on things, is that I realized that you can actually change your microbiome. Now, in this case, the study showed that, or it was making the claim that this microbe lets the digestive system do a better job of, I think, pulling carbohydrates out of seaweed. That if you don't have this microbe, you won't be able to digest it. It'll just go right through. Whereas people who do have this microbe are able to, fully utilize all the, more of the nutrition that's available in seaweed. So that's kind of cool that you can manipulate through the kinds of environment that you expose yourself to. You can manipulate your body's ability to be able to digest certain kinds of food. And that set me off in a whole bunch of different directions that I've been intrigued about ever since. The fact that you can change something about your microbiome that has a direct effect on the rest of your digestion. It's kind of cool.
How old were you when you were living in Japan?
I was in my 20s. So
Gotcha.
I moved there right out of college, and I lived there for most of the next 10 years. Yeah.
That's cool.
So it was
I
not when I
only
was an infant. It wasn't when I was a child. It was, I
right.
was an adult.
Right. I think that's interesting because there is this idea in the literature of the critical window of opportunity, or a time when, if you don't kind of lay the foundation for the microbiome, that modulating it later down the road becomes difficult. How true do you think it is? To what extent might there be a critical window of opportunity? And if someone, for some reason or another, does not get a robust microbiome in their early years up until 2 years of age, is there still hope for them to still be a healthy person? Can they still remedy that somehow?
It is pretty clear, and I know this from my own experience, is that you can manipulate your microbiome. So you can do things to yourself based on what you eat. Certainly antibiotics. There are even more desperate measures like FMT where you can manipulate what kind of microbes you have. And this thing about the seaweed microbe was kind of a proof for me. Um, so now, are there critical periods where maybe it matters more than other times in life? I think it's pretty clear that infants, especially when they switch to eating solid food, it's a pretty obvious change that happens. And I've seen this myself with results that I trust from people who've sent me their own microbiome samples, and you can see the difference between like a mother and a child. Another interesting thing about it was that I was very intrigued when I looked at results from mothers and their babies, that we'd find out that the baby was a complete subset of the mother's microbiome, which is kind of interesting, and not a complete subset of the father, which is kind of a interesting observation about stuff. But clearly, something changed when that baby switched to using solid food. So there is a period of time when a major shift happens. Now, if that baby was somehow exposed to the wrong things or didn't get exposed to the right things, we know there are plenty of examples of intriguing changes that seem to happen that result in autoimmune diseases or whatever because of a person's not exposed to certain kinds of microbes. We know all that stuff. Um, there's the hygiene hypothesis. There's the like what's called the old friends hypothesis. There are different ways of being able to express the idea that you need certain microbes at certain times, and if you don't get them, bad things happen. I don't think we know a whole lot about the details of it because for each one of these things, there are also plenty of exceptions. Um, I'm kind of an exception. One of the things that another part of sort of an intriguing thing to me is when I was 5 years old, I was I was given an appendectomy, and for no really good reason. It's just because the doctor was thought that that was a good idea. And as a result of the appendectomy, I was given tons and tons of antibiotics. So for like probably close to a year of my early development years, like between ages 5 and 8, I was given tons and tons of antibiotics. And you would think, based on everything, that that would mean that I would end up with all kinds of autoimmune diseases, because there are plenty of examples of people who end up with allergies or other kinds of problems. And I don't really have those kinds of problems. So I'm either an exception to the rule or something else. One of the intriguing ideas that was presented to me is maybe for whatever reason, I ended up getting a microbiome that was because it had been exposed to so many antibiotics that it developed mutations that made it more robust than normal. Like that's possible too, and maybe I just was one of the lucky ones. Um, so who knows? It's just really hard to tell some of these things.
Definitely. I think the temporal dynamics are really interesting. And, there is this concept of resilience. And I think some microbiomes are more resistant to assault like antibiotics than others. But I think,
Yeah.
especially considering the fact that some more recent research suggests that the appendix may actually not be a vestigial organ and maybe
Yeah.
might even serve as a safe house for beneficial
Yeah.
bacteria. There's this hypothesis that after a bout of food poisoning or after a cholera infection, bacteria repopulate the large intestine from the appendix. So that's a really intriguing hypothesis that,
Yep.
maybe it's not vestigial after all. Maybe it serves some purpose. And I
Yeah.
just
I
feel
think
like
that's a good example of it. Yeah.
all this sort of emerging evidence points to the lesson that the absence of evidence is not evidence of absence, meaning like just because we can't
Yep.
find a function for something doesn't mean that a function doesn't exist. And this also reminds me
Yeah.
of a recent discovery surrounding the brain. And like we found out that the brain has lymph nodes in just 2015, and this actually
Hmm.
led to the creation of a new term, the glymphatic system, which refers to
Hmm.
the immune component that does exist in the brain. And
Hmm.
when this famed neurologist Ramon y Cajal couldn't find evidence, we assumed it wasn't there, but then we found out that that wasn't
Hmm.
true. And you do write a little bit in your personal science ebook about how the brain isn't sterile and the implications that it has for
Hmm.
Alzheimer's disease. Could you talk to us a little bit
Hmm.
about what you think the pathogenesis or the origin of Alzheimer's might be? And like, should we be moving away from this beta amyloid model of the disease, that one that embraces the microbial aspects as well?
Yeah. So again, I'm not an expert, and but I think of myself as a reasonably intelligent person who's able to look at different things, and maybe partly because I'm outside of the specialty, I can look at things with maybe slightly fresh eyes. So take this all with a grain of salt. But it is intriguing to me that Alzheimer's, like a lot of things, is might be an infection of some kind. And one of the most like compelling pieces of evidence that I've heard is that an early symptom of Alzheimer's disease is loss of smell. And to me, that maybe indicates that there's something weird going on in the nasal microbiome. And so, like, kind of a theory that I have, again, based on nothing other than just speculation, is that maybe there are these microbes that we can become infected with. Maybe some people are more likely to be infected either because of their environment or their genetics or who knows what. And they're carrying this microbe that lives in the nose for a long, long time, and it uh, somehow is able to defeat the human, like the immune system, maybe because it provides a benefit to something else. So maybe these people live their whole lives having fewer colds than other people. But it's because there's a microbe that's sitting in their nose that's protecting them from getting bad colds. But when it tips over at a certain age, it makes its way into the brain and it beats back whatever immune system there is that's protecting the brain. And now it starts to form all kinds of bad stuff in the brain. And that's what we see as Alzheimer's. Like I could see that as being a potential like a research direction. And I bet there are a lot of similar things like that. There's this one really weird microbe that was discovered in, I think it was Japanese people, um, where it was discovered that I think it was sexually transmitted from, but it occurs in like infants or very young children. And they discovered that these people didn't have any kind of problems at all until later in life, and they would develop a form of leukemia that's very rare in old people. And they were able to trace it to this microbial infection that they'd got when they were, young. And I bet there are a zillion things like that where we are harboring these infections that are just kind of sitting there, waiting in the body, maybe protecting us from something else for a while until they go off the rails and cause some other kind of problem. And so I
Definitely.
bet there are a lot of examples like that. Yeah.
Agreed.
Yeah.
Yeah, I think a lot of the stuff that we're able to tolerate in our younger years, maybe when our immune system is robust, maybe when you're older and maybe more immunologically frail, that could start causing a problem later down the road. I do think it's interesting, like the idea of the nasal microbiome, because, that potentially can help with nitric oxide production, which also exerts some
Hmm.
antimicrobial effects. And I think there were some research institutions that were looking into nitric oxide treatment to kind of help with the alleviation of COVID-19 symptoms as well. They were kind of using that as, I think, an
Uh-huh.
intranasal therapy of sorts, perhaps. But if it
Yeah.
is
Interesting.
true that,
Interesting.
oh, I was just saying, if it is,
I was going to say, like,
mhm,
if I can, so maybe we're saying the same thing here, but uh, these it if you believe that there is some kind of an infection going on, it might lead to a couple of possible things that you might want to think about for either treating yourself or preventing yourself. And some of them that I found intriguing to think about are we know that the microbiome changes based on environmental changes. So just like me living in Japan, or it's pretty clear that when you move to a new part of the country, or you adopt a different kind of diet, you'll see a change, a noticeable change in the microbiome, and that happens all the time. Same thing goes with just like me eating, drinking the kefir, or there are lots of other things that affect the microbiome. Some of the things that are maybe less obvious that may affect things, that if you're having problems or you want to shift, you want to get yourself into a slightly different state so that maybe you can shake off some kind of problem that you're facing, or maybe you want to improve things a little bit. A couple of things to think about are think about ways that you might change your microbiome. So, in the example of the nasal microbiome, um, maybe you can expose yourself to different kinds of odors, like perfumes, different kinds of soaps, different kinds of things that cause, that might affect the nasal microbiome. Maybe those are ways that you would change the way that the microbiome works in your nose. A big one that I like is fasting. I run into people all the time who have gut issues and they've tried everything. And I encourage them, and a lot of times I'll ask them, it's like, when is the last time that you went more than 24 hours without eating anything at all? And often it'll be the case, and I'm surprised that for most people, they'll say, I don't remember I've ever gone 24 hours without eating anything at all. But that, to me, just it means that you've got some microbes in your body that know that they can count on getting food at regular intervals. And those probably are optimized toward sucking up all the nutrients that come out of that kind of food that you're getting regularly, at the expense possibly of other microbes that might be good for you, that might change your health somehow, for maybe for the better, that that can't get a bite because they're constantly getting outcompeted by this thing that is expecting food all the time. So if you just go without food for a little bit, fasting for a day or two, maybe that's enough to lower the levels of those kind of microbes, so that now suddenly the ones that um, are more hardy, that don't need to have food all the time, that don't depend on that, maybe they can grow a little bit better. And so that's an example of the kind of intervention that I think you can think about if you're thinking about how the microbiome might affect the body. It's like, think about ways that you can change your environment a little bit in ways that might shift the types of microbes you have. And if you're trying to get out of a particular state, think about things that you might do to just change the way that your microbiome works.
I like that approach, and I do feel like the constant availability of food can present a lot of problems just based off of the necessity of having fasting periods. And I feel like this kind of goes hand in hand with other effects that fasting produces. Like you're saying, there are microbiome shifts, and some research suggests that like Akkermansia and Lactobacillus will increase if you lengthen your fasting windows. But then there's also the matter of
Hmm.
just things like autophagy and like GI cleanup.
Hmm.
Like there's this set of peristaltic waves in the gut called the migrating motor complex, and that takes a number of hours to sweep through the small intestine and kind of like get rid of food or any sort of debris, mucus cells that are gotten stuck to the lining. And I feel like a lot of these things just aren't given the opportunity. It's sort of like, if there is a street cleanup crew, it can't do its job while there's cars on the road. And I hear that analogy quite a
Yeah,
bit.
yeah,
But then, there's also this
yep.
matter of autophagy. And like you're saying, if there is always nutrient availability, we actually have some nutrient sensors, like mTOR, for example, and they sense when there is some sort of food, when food is no longer available. And a lot of these mechanisms, they're just not ever turned on unless you are in a fasted state. So a lot of normal physiological processes are not happening if we are constantly eating around the clock. So I think that's a great
Yeah.
thing to be
Yeah.
mindful of.
Are you familiar with Seth Roberts? With he was a Berkeley psychologist for many years, and he kind of founded the idea of at least for me, the idea of personal science. And he wrote a popular blog until he passed away like 6 or 7 years ago. And he had this idea that, first of all, he was a very interesting, creative guy, and he's worth reading about just because he had a lot of different perspectives on things, and he's been very influential to me. But he had this idea that we have our bodies have a natural set point where it's expecting certain types of nutrients and certain types of foods, and whenever we get out of that set point, things, the body naturally wants to get back to that set point. And he thought that there's this way you could change the set point, and the way he said was through having flavorless calories. So he would encourage people to like his he called it the Shangri-La diet, where the idea is drink. This is all you have to do. You can eat whatever you want. You can get as much exercise. It doesn't matter. Nothing else matters other than every morning drink 3 tablespoons of some kind of flavorless oil, probably like something like olive oil, and that's all you have to do. And if you do that, you'll lose weight. And a surprising number of people lose weight over this diet. And that's why it's just incredible results. And he said the reason was because you would, by consuming flavorless calories, your body would shift where it was expecting to get like normally when it gets food, it thinks, oh, I'm getting food. I'm going to prepare myself and expect to have a certain amount of leave a certain amount of it as fat, use a certain amount of energy, and if I just sort of expect certain things. But when you give it flavorless calories, it doesn't know what to do with it, and so it's like, oh, okay, well, I'm gonna take this in, and because the other part of the body says, you know what, I'm kind of full. This high fat that I just drank is olive oil. I'm already full, so I'm not going to eat anymore, and just naturally your appetite goes down, and you kind of fool your one part of the body at the expense of the other body and use that to change that set point. And over time, that set point actually can change. This was his theory. And now suddenly you will no longer need the olive oil. Your body will now expect few, it'll expect the right amount of calories for you, and you just simply won't eat as much because you won't feel like it. And I think there's room for a lot more other of those sorts of ideas. And I bet a lot of us, especially like people who are healthy or people who have defeated certain diseases. I bet a lot of us have just kind of stumbled onto those kind of little quirks, and fasting is just one of them. But I bet there are a lot of them. If you just look around and you look at people who are successful at fighting different kinds of diseases, they probably just do something little stupid little quirk that is affecting the microbiome somehow, and and just being mindful of what you do and asking yourself all day long, am I doing this because I am just doing it just out of habit, or is there some good reason for why I should do it? And maybe I should change. And like that level of curiosity, that's what I think is the core part of personal science, is like to always be like curious about, is there something that I'm doing that that maybe if I change a little bit, I can change the course of my like my health. And so I think that sort of level of just being aware of things is just super helpful.
Definitely. I feel like that idea of flavorless calories, it kind of reminds me of the fasting mimicking diet that Walter Longo has conceived of. But the idea of of having a set point or almost like this thermostat in the body, I think this kind of would inform a lot of yo-yo dieting that often observes where, people they might be able to lose weight initially, but it very quickly comes back on. The body's wanting to return to that set point. So it's almost like unless you reset what that set point is, it's hard to have long term success with any sort of endeavor. I think you did sort of do some metabolic experiments of yourself with yourself as you were kind of figuring out the associations that you saw between continuous glucose monitoring and the microbiome. Can you talk to us a little bit about those experiments?
I had a lot of experience with that. So a continuous glucose monitor is really fun. And have you tried one? Have you used it yourself yet?
I
Or
have not.
have you tried? Oh, you've got to do it because it's easy, it's cheap. I buy mine for about $30, $35 at Costco, and it lasts for 2 weeks. And you just use a regular iPhone, and you can go see what your glucose levels are at any time, so you can immediately see how it affects your like your diet, or your you can immediately see the effect that the food has on your glucose levels. And I discovered a bunch of things. Actually, let me see if I can show you. This was a fun one that I found out. I always heard that oatmeal is good for you, and so I would eat a lot of, I'd eat oatmeal. But I discovered that when I eat oatmeal, it causes this big, huge spike. So this green line here is when I have a bowl of oatmeal, just plain, without any milk or anything else in it. The red line is when I have it with milk. Generally speaking, people say that having higher glucose spikes is bad for you. And so you want to kind of keep it relatively on the low side. And I was able to see for myself that just adding a tablespoon of butter to oatmeal was able to give me a much more steady, glucose level. And same thing happened when I looked at pasta. So here is this was whole wheat pasta, the green line here. And again, higher is worse. So you want to have it be more steady. And I found out that by having pasta with pesto, it keeps my glucose levels more in check. There have been lots of studies that have been done on this sort of thing, where it turns out that depending on the kind of microbiome you have, this is a very famous study that was done by a lab in Israel, where they were able to show that for some people, eating plain white bread was actually better for them than eating whole wheat bread, which most of the time we think that that's a good thing. And here's another study that was done by Viome, another microbiome company, that shows that there's some people react differently to like a banana. So in this case, somebody who ate a banana had a very high glucose response, versus when they had sprouted grain bread, they had almost no response at all. And then vice versa. Another person was exactly the opposite. And again, this is like a key part of the principle of personal science, is that your reaction to things might be very different than whatever the study says is the average on stuff. And so I've been very intrigued by this. Some of these ideas that I got from doing my CGM experiments were based on the microbiome testing that I'd done, which suggested that you should try this or that. And that's why both Day 2 and Viome provide this sort of testing that's been correlated with glucose response, and they're able to show that certain types of foods actually are better for you than other kinds of foods. And that's something that's been very intriguing to me. I found out a lot of interesting things about myself through that.
In your oatmeal experiment, was it whole milk that you added to see if it would produce a smaller glucose
Yeah,
spike? Exciting. Yeah,
yeah,
I only asked
hmm
because, whole milk
hmm
has that fat component, and typically fat tends to prevent
yeah,
glucose spikes compared to the more insulinogenic protein. And of course, carbs are the most
yeah.
likely to cause spikes. So I guess that would also
Yeah,
kind of
that's
be
right.
in
Yeah.
line with what you would expect to observe when you're adding butter, because that is a source of fat. So, also, I think
Yeah.
that kind of speaks to the importance of maybe combining foods or like eating your carbs clothed instead of naked to kind of help
Yeah.
prevent that spike.
Yeah.
Did you?
One of the other things that, like, other nice thing about a CGM too is that, which is why I encourage people to wear one for a while, is that, or for at least, try it for 2 weeks or a month or something like this, is that it also helps. It put me kind of more in tune with what it feels like to have a lot of glucose. So you're able to see in real time that your glucose level is high, and you get a sense of what that feels like. And then after that, even without having the monitor, now I'm able to taste certain kinds of food and see what happens. And then I feel like I know that my glucose is high. And it just kind of trains your body to be a little bit more observant to that stuff, which I think is definitely worth doing.
So perhaps these experiments
You
have
definitely have
helped.
to try CGM.
I will definitely. Would you say that that experimentation has helped you develop a more astute sense of interoception there, where you're more kind of in tune with what's happening in
Yes.
your
Yeah.
body?
Yeah.
That's
Yeah.
exciting.
Yeah. Same thing goes with, like I'm a big fan of wearables, and like I have an Apple Watch that I use to measure things, and again, it's I think what it is really useful for is mindfulness. Makes you more aware of what you're doing, and I, over the years, have become less interested in this idea that, oh yeah, if I collect enough data, I can like super optimize myself, and I'll, find out that exactly what, how much I should do for this and that, and that's what you would think like if you were a computer programmer and you were thinking that the body is just another piece of hardware that you can manipulate somehow through software. It's way more complicated than that, but one of the one of the nice things you get by wearing, devices, or having a CGM, or diagnostics like the microbiome testing, is it gives you a little more sense of who you are and how you react to things. And I think that's the important thing. So I think just being like more mindful about what you're doing and being a little more deliberate and opening your eyes a little bit more to what sorts of things affect you and don't affect you, that I think is the secret. I think it's yeah, that's a powerful thing that you can develop for yourself. Just the
So
mindfulness.
definitely. And it seems like the benefit of self-knowledge, yeah, can't really be understated either. Just the more you know about yourself, the
Yeah.
more you can.
Yeah. And it's surprising how many people go through life just doing stuff because, for whatever, they just, that's what they do, and not really thinking about, like, why are you doing it this way? And is there
Right.
a better way to do it? And just be a little bit curious. And, people who have, they're just like really annoying pathologies. I'm thinking of like like migraine headaches, or people with various different gut problems. They'll just kind of accept the fact that that's who I am, and that's just something I have to deal with, and not really think like, well, maybe there's something simple you could be doing that might manipulate this and change it for the better. And sometimes it takes a serious pathology, like you get floored by something really bad and maybe hospitalized, or some terrible thing, to jolt people out of that. But I think that all of us can benefit by being, thinking a little bit more proactively about just being self-aware about who it is you are, what you're doing, how you might be doing this. So why do you think that this is good or bad for yourself? And just being like that level of curiosity and skepticism about things, I think is just a really, that's just a powerful, maybe even more powerful way to solve a lot of health problems than just collecting a lot of data.
Agreed. Yeah, I do think that the systems that you develop and just the problem solving skill is often like a much bigger thing to be gained rather than insights from the data itself, because that's just so transferable. And I love that idea of like maintaining the skepticism and really questioning, why is it that we're
Hmm.
doing what we're doing? And am I eating the way I eat because of what the food pyramid tells me, or because of my plate recommendations? Or
Hmm.
am I eating this way because it's in line with what works best for my specific needs? And this is the way that maintains my
Hmm.
metabolic health. So I do feel like that data has the potential to really
Hmm.
inform our choices and help us make much better decisions. What was the most
Hmm.
surprising result from your personal experiment? Something that you did not expect to see when you were maybe tracking your fitness or sleep or your glucose levels? Was there anything that really stood out as, I was not expecting to see this relationship?
Well, I guess that happens all the time where I'll find something that I wasn't expecting. Oh, here's one. Alcohol, particularly wine. Uh, I was not expecting that in my case, drinking alcohol significantly lowers my glucose levels. And for some people, this is a sign that it's a healthy thing. And I don't know, you're like your mileage may vary, and so you should look at this yourself. But in my case, I was able to show with doing various side-by-side experiments. If I have, say, after dinner, if I have a bowl of ice cream for dessert, my glucose will shoot up like you wouldn't believe, like as you would expect. If I have a glass of wine after dinner and I have a bowl of ice cream, my glucose doesn't go anywhere. And that is so non-obvious. um, when I looked into this more, and I discovered that there are actually some people, and apparently I'm one of them, where this is a known characteristic. It's not necessarily a good thing because what's happening is, and this is how I explain it, my body sees alcohol as something that's this deadly poison. And it says, oh my God, all hands on deck. We got to get rid of this poison and forget about worrying about what we're going to do with the glucose for now. Let's just, pack it away. Let's get rid of this alcohol first. And so we're going to put every single, cell that we got in the body and trying to get rid of that alcohol before we worry about the glucose. And so it's not like you've solved your glucose problem. You've just put it off for another day. But that's an example of something that I would never have guessed was true about my metabolism.
I can imagine. Yeah, that's a really interesting result. And I do feel like your mileage varies a lot just depending on the way that alcohol is kind of metabolized for you specifically. I mean, of course it's a zero order process for everyone, but still there's still differential downstream effects after that really basic like liver processing because even that, the CYP450 liver enzymes differ for people. So it's not as though
Yeah.
our biochemistry is identical the way modern medicine would perhaps assume. But I really love that redemptive message that anything we're dealing with, it is perhaps reversible or at least something that can be improved upon with the right knowledge and with the right data. And I really appreciate you coming on and sharing your insights with us. Thank you.
Yeah. Yeah. I would suggest people go to personalscience.com because that's where I'm going to be starting to really like writing down more of my because otherwise, please follow me on Twitter. My last name's Sprague. Is where I try to tweet, every day or two about something related to my personal science work. And I'm always eager to have people talk to me. So like if you come up with something interesting or if you see something interesting or whatever, just please contact me. I always encourage people if they get microbiome results and they're looking for another pair of eyes to look over the results. I'm happy to do that. So and to find out my contact information, you can go to richardsprague.com
Wonderful. Thank you again
and
so
see
much
how
for
to
your
contact me
time.
directly
And please
there.
come back soon. Awesome sauce.
Thanks. It's great talking to you, Nita.